Breaking news. Recent gene therapy breakthrough in 2023 has made it so those with a rare type of complete color blindness to faintly see red. In the test, people with total color blindness were able to distinguish red from a gray background after undergoing gene therapy. This process is done by injecting a virus that carried the right gene into the subretinal area, where the cone cells of the eye are located. The virus corrects the defective genes with the new gene, and after a few months, the participants were able to see a slight red against gray or black backgrounds. Although not much has progressed since or before, this is definitely groundbreaking for color blindness, and hopefully we can see some improvements in the future. But what is color blindness, and how does the mutation work? We must look back to the DNA of the cells in the eyes. First of all, color blindness is most simply when you are unable to see colors in a normal way, such as the inability to distinguish between different colors, or seeing two different colors at the same time. This happens when cones, which are nerve cells in your eyes, they aren't working properly, as they are what allow you to see color. These genes have genetic code that send instructions to the cones for them to function. And when these genes go wrong, due to colorblind mutations in the chromosomes, the cone's functions are affected. The main kind of color blindness is red-green color blindness. And color blindness is almost always inherited, passed on from the mom or dad's X chromosome. Once you have this, you'll have it for life. There's no treatments or cures available at the moment. And while color blindness may seem insignificant, it can actually be life-ruining. For example, professions like graphic design are likely a pipe dream for those who are colorblind. Or people who are driving would likely be not allowed to be on the road, as it would be very dangerous when you can't differentiate between the red and the green light. Here's a slideshow presented by Amir that shows the steps of the mutated protein synthesis.
Thank you so much, Albert. Hello, everyone. Today I'm going to explain how a small change in our DNA can lead to something called red-green color blindness, where people have troubles telling reds and greens apart. Well, right off the bat, I'm just going to start with the DNA mutation. The gene responsible for red-green color vision is located on the X chromosome, commonly affecting the OPN1LW, which is the red pigment gene, or OPN1MW, which is the green pigment gene. A mutation might be a point mutation, deletion, or gene rearrangement. In some people, a small mistake or mutation happens in these genes. For example, one letter in the DNA might change, like an A turning into a T. This is called point mutation, for example, and it can stop the gene from working properly. Next, I'm going to talk about transcription. Copying the gene, the other name for it. Inside the cell's nucleus, the DNA is copied into mRNA through a process called transcription. The mutant gene is transcribed into messenger RNA or mRNA. The DNA sequence with the mutation is copied into mRNA. If the gene is mutated, the mRNA copy carries the same mistake carry on. Sometimes, this mutation causes stop codon to appear too early, cutting the protein short. Next, I'm going to briefly talk about the translation aspect, which is the building the protein part. Then the mRNA goes to the ribosome from here, where it's used to build the protein. This is called translation. Ribosomes read the mRNA sequence in sets of 3 bases, or the codons. Transfer RNA or tRNA brings the corresponding amino acids. The sequence is translated into protein. In this case, upsin for our color blindness that we've chosen. The pigment in cone cells. Upsin helps our cone cells in the eyes detect colors, but if the mRNA has a mutation, the upsin might be incomplete or faulty, mistaken. Protein folding and the problem that comes with it at the end of the day. The upsin protein folds into a specific shape to function in the eye's retina. A mutation might cause either misfolding of the upsin protein or non-functional protein, or the inability to absorb light at the correct wavelength. But once it's built, the upsin protein needs to fold into the right shape. If it doesn't, well, it won't work. A broken upsin protein means cone cells can't detect red or green lights correctly or apart from each other. And last but not least, the result, which would be color blindness. If the pepsin protein is non-functional or not produced, then this causes red-green color blindness, where people mix up red from greens and they can't really distinguish between them. And a few fun facts that I'm just going to quickly go over is, what does it even look like, if you guys are wondering? Here's a picture of what someone with red-green color blindness might see. Well, who usually gets it is more common in boys than girls because the gene is on the X chromosome, and since guys have only one X, a single mistake can cause color blindness, so it's more potential for males to carry this. And the last one is some animals like dogs are naturally colorblinded, which was kind of interesting for me to know, and this is basically how they see the world. Thank you so much for listening.